Okay, let's dive in. The really surprising thing, maybe the most surprising, about the DNA double helix discovery,
What's that?
it's that Watson and Crick, well, they didn't actually run some huge defining experiment themselves.
Right, yeah.
Instead, they cleverly pieced together existing findings. Some of them even seemed contradictory at the time. It was almost like a new way of doing science.
It really was a synthesis. And you have to remember, back in the early 1950s, scientists were, well, pretty sure DNA held the genetic secrets, not protein.
Right, the focus had shifted.
But knowing it's important and knowing how it works structurally are two different things. Nobody knew its 3D shape. How was it all put together? That was the big question.
A huge gap in understanding.
Exactly.
Yeah.
And you had Linus Pauling, a giant in the field, proposing a 3-stranded helix with the phosphates kind of tucked inside.
Okay, a triple helix.
But then there was Rosalind Franklin's X-ray work, especially that amazing photo 51. It just screamed double helix with that distinct X pattern.
So, conflicting evidence right there. Pauling's idea versus Franklin's picture.
Precisely. How do you build a model that fits the X-ray data, obeys the laws of chemistry, and explains how DNA actually stores and copies information? That puzzle, that scientific tension, was the real driving force for Watson and Crick.
Got it. So that's what we're exploring today, how they took on that challenge. We'll look at the key clues they used, like Franklin's data, let's call that figure 1, and Chargaff's rules, figure 2 maybe, and see how they built up to that final famous double helix.
And what's so fascinating, as you said, is they didn't start with lab coats and test tubes for this specific problem.
No, what did they do?
They took this really innovative paper and model approach. They looked at Rosalind Franklin's measurements from her B-form DNA X-rays, the precise numbers like the 34 angstrom repeat, the 3.4 angstrom rise per base, about 10 bases per turn.
So not just the picture, but the actual dimensions.
Exactly. They treated those numbers as hard constraints, like architectural specifications for the molecule.
Okay, so they had the specs. Then what?
Then, and this is the cool part, they started building physical models, like actual models.
You mean with their hands?
Yeah, literally, using things like cardboard cutouts for the bases and sugars, metal bits representing atoms. They were physically trying to fit these pieces together.
Wow.
You can picture them sort of manually twisting and turning these parts, trying to see if they could get a consistent width, aiming for about 10.5 angstroms for the base pairs.
That's such a hands-on, almost playful approach compared to just, I don't know, complex calculations.
It was a huge departure. It let them visualize things, test configurations intuitively.
Yeah.
It really shows they were willing to embrace a different kind of thinking, more physical, more tactile, to crack this molecular code.
Okay, so model building was key. But what about the chemistry? You mentioned Chargaff's rules.
Right. Erwin Chargaff, that was the other huge piece. His biochemical work had shown something really consistent and, frankly, weird about DNA.
Weird how?
In any DNA sample, the amount of adenine, A, was always pretty much equal to the amount of thymine, T, and the amount of guanine, G, was always equal to cytosine, C.
A equals T, G equals C. Always.
Was. This AT-GC relationship, sometimes called purine equals pyrimidine, was this other crucial fact that any correct DNA model just had to explain.
Okay, so they're playing with their models, trying to fit Franklin's dimensions, and they know AT and GC. How do those two things connect?
That's a stroke of genius. Inspired by Chargaff's ratios, Watson and Crick proposed it wasn't just about amounts being equal. They suggested A specifically pairs with T and G specifically pairs with C, like puzzle pieces.
Okay, specific partners?
Yes, held together by hydrogen bonds. And this specific pairing was elegant for two big reasons. First, an A paired with a T has almost exactly the same width as a G paired with a C.
Oh.
So this pairing naturally creates a helix with a consistent width all the way down, roughly 20 angstroms across, which perfectly matched the X-ray data.
Boom. So the physical shape from Franklin and the chemical rules from Chargaff suddenly snapped together perfectly. That's amazing.
It really was a eureka moment, but it wasn't just about fitting the shape.
No. What else?
Well, the second reason, maybe even more profound, was what this pairing implied.
Which was?
It immediately suggested how DNA could be copied if you could just unzip the two strands.
Unwind the helix.
Exactly. Then each single strand could act as a template. You just bring in the matching partners, A finds T, G finds C, and build a new complementary strand along each original one.
So ATGC becomes the rule for copying.
Precisely. It was this incredibly simple, elegant mechanism for replication baked right into the structure itself.
Mind-blowing. And they also figured out where the different parts went, right? The backbone and the bases.
That's right. It wasn't random. They positioned the phosphate sugar backbone, which carries a negative charge, on the outside of the helix.
Okay, why outside?
Well, a couple of key reasons. First, it makes sense chemically. DNA lives in water inside cells, and having those negatively charged groups on the outside explains why it behaves as a polyanion, a molecule with lots of negative charges at normal pH. It also allows it to interact with positively charged proteins.
So it fits the cellular environment. Makes sense.
Exactly. And crucially for their model building, putting the backbone on the outside produced an X-ray diffraction pattern that looked remarkably like Franklin's B-form image.
So it matched the picture again.
Yes. And it solved a big problem with Pauling's model. He had the phosphates on the inside. Chemically, that's really problematic. All those negative charges crammed together would repel each other strongly. Watson and Crick's outside-in model avoided that.
So their structure wasn't just geometrically right, it was chemically stable too. Got it. And then there's that famous line in their paper, almost hiding the biggest implication.
Oh, it's classic understatement, isn't it? The very last sentence of that short 1953 Nature paper. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material.
Just casually dropped in there.
Almost. But that single sentence, it contained the key to heredity. It pointed directly towards semi-conservative replication, the idea that when DNA copies, you get two new helices, each with one old strand and one new strand.
Flowing directly from the ATGC pairing.
Directly. So the real innovation wasn't just finding a structure, it was finding the structure that integrated the chemical rules, Chargaff, with the physical constraints, Franklin, and immediately revealed its biological function storing and copying information. It was the whole package.
Okay, so we've seen how the model came together. But let's talk about the innovation itself, because as we said at the start, they didn't actually do the core experiments on DNA structure.
Right, and that's really important to grasp. Their genius wasn't in generating new raw data here. They famously did no direct experiments on DNA structure themselves.
So where was the innovation?
It was in the synthesis. This incredibly creative act of pulling together existing knowledge from really different areas. You had Franklin's physics-based X-ray crystallography. You had Chargaff's meticulous biochemistry on the base ratios.
Different fields entirely.
Totally. And they even learned from Pauling's attempt. His incorrect model showed them what not to do, what pitfalls to avoid, like the phosphates in problem. They weren't bound by this idea that discovery only comes from new experiments.
They prioritized the model.
Exactly. Building a model that fit all the known facts, using intuition and, well, that physical model building we talked about. It was like molecular visual thinking.
And the place they were at, the Cavendish lab, that must have helped.
Oh, hugely. The environment there was critical. People like Perutz and Bragg encouraged model building, theoretical thinking. It wasn't just about experimental results. So even though Watson and Crick weren't officially assigned to DNA initially, they had the freedom and the intellectual climate to pursue it.
And they weren't totally isolated. Information was flowing.
Absolutely. They needed Franklin's data from King's College. They needed Chargaff's ratios from Columbia. Even Pauling sending his manuscript beforehand, while it was wrong, was useful information. It spurred them on, actually.
It sounds like this intense period of discussion and integration, picking the right threads.
Intense is right. The famous discussions between Watson and Crick themselves, arguing, debating, refining ideas, even down the pub, apparently, it highlights how dynamic collaboration works. And this approach, focusing on synthesis, on fitting existing data into a coherent model, it wasn't necessarily the mainstream way back then.
Really? How so?
Well, in many fields, the big emphasis was on generating your own original experimental data first and foremost. Watson and Crick showed that a massive breakthrough could come from, cleverly connecting the dots that were already there, maybe just in different places, reinterpreting and integrating existing knowledge.
That's a powerful idea.
It really validated that synthetic approach to science.
So wrapping this up, for you listening, especially if you're diving into complex biology problems, what's the big takeaway here?
I think the story of the double helix is just such a fantastic reminder that huge breakthroughs don't always come from one single giant experiment. Often they come from making these insightful connections between bits of knowledge that maybe seemed separate before.
Seeing the bigger picture.
Exactly.
Yeah.
It encourages you really to look at problems from multiple angles. Don't be afraid of different methods, maybe model building, maybe computational work, alongside traditional experiments. And be open to finding clues in unexpected places, maybe from different fields entirely.
So keep your eyes open for connections.
Definitely. Think about the things you're learning, the challenges you're facing in your own studies or research. Are there pieces of information floating around, maybe seemingly unrelated, that could actually hold the key if you just put them together in a new way?
That idea of synthesis, connecting the dots.
That's it. How could that approach help you find your next aha moment? It's about looking beyond the obvious experiment and integrating everything you know to see the underlying pattern.